Hi everyone, it's Dr. LeBlanc with Unit 6A, the regulation of eukaryotic gene expression. In this video capture lecture, we're going to discuss transcription factors. Our learning goals are to understand the actions of general and regulatory transcription factors, to understand how the mediator complex works, to understand the interaction of transcription factors and DNA, to understand the interaction of transcription factors and small effector molecules, and to understand the mechanism of steroid hormone receptor gene regulation. General or basal transcription factors are required for RNA polymerase to bind to the core promoter, TF2D and the Tata binding protein, for example, and for transition to the elongation phase. TF2D is a general transcription factor or a basal transcription factor that recruits RNA polymerase to the promoter. Here we have TF2D bound to the core promoter, and it is recruited there itself by activators, transcription factor activators bound to coactivators. They bind in an enhancer region. In addition, we could also have repressor proteins bound to silencers in the DNA that prevent TF2D from binding. The regulatory transcription factors, those activators and repressors from the previous slide, can respond to developmental and environmental signals. They influence RNA polymerase and general transcription factors. They turn genes on or off and can modulate the rate of transcription. They bind to DNA cis elements called silencers or enhancers. So the enhancer is the segment of DNA to which an activator protein or a transcription factor will bind. When activator proteins are bound to enhancers, the core promoter is occupied by all the transcription factors it needs to get RNA polymerase to transcribe a gene. If, however, a repressor protein is bound to a silencer, or RNA polymerase is not recruited to the core promoter or is prevented from elongating the gene, and transcription is inhibited. So if we look at a schematic representation of a eukaryotic gene, we have the coding region here. This would be the start site of transcription. We have our Tata box, and we have another proximal promoter region that contains other binding sites for some of these other general or basal transcription factors. At some distance away, we have enhancers that will bind activators. We could also have silencers that bind repressors. But because of this great distance, the DNA has to fold around so all of these factors can interact. What are some of the structural features of regulatory transcription factors? How do those proteins bind to the DNA? And what are some of the other domains? They have a DNA binding domain. They also have dimerization domains, and they have binding sites for effector molecules. Interestingly, some of the domains, especially the DNA binding domains, may serve as either those, DNA binding, or in some cases, dimerization domains in other proteins. For example, zinc fingers can be either a DNA binding domain in one protein or a dimerization domain in another protein. So here are some of the domains that bind to DNA. Helix turn helix, and then there's another domain called the helix loop helix. Notice that this is a dimer, 2 polypeptides. Zinc fingers, which bind molecules, atoms of zinc, and leucine zippers that have hydrophobic stripes, which allow the polypeptides to, these alpha helical domains in the polypeptides to coil around each other, called a coiled coil. Let's take a look at the zinc finger domains.
Zinc finger domains are structural motifs used by a large class of DNA binding proteins. They use centrally coordinated zinc atoms as crucial structural elements. The zinc atom is coordinated by 2 cysteine residues from the beta sheet and 2 histidine residues from the alpha helix. A single zinc finger domain is only large enough to bind a few bases of DNA. Zinc fingers are often found in tandem repeats as part of a larger DNA binding region. The helical region of each zinc finger rests in the major groove of the DNA. Amino acid side chains project out from the helix and contact bases in the DNA. The identities of these side chains determine the precise DNA sequence recognized by each zinc finger. Assembling different zinc finger motifs allows precise control over the sequence specificity of the protein.
So the mediator is a protein complex that acts as a go-between for transcription factors. The mediator complex binds to activator proteins that are bound to enhancer regions. Notice that the mediator complex bridges this long distance in between enhancers and the core promoter. When a mediator complex has bound to an activator protein, it releases RNA polymerase, and RNA polymerase can then transcribe the gene. This is its active conformation. Other times, the mediator complex can bind repressors that are bound to silencers. When this happens, the mediator complex sequesters RNA polymerase and prevents it from transcribing a gene. So repression happens when the mediator complex binds a repressor protein. Here's another diagram indicating that activators bound to enhancers, several different activators or the same activators bound to repeated enhancers that are at great distance away from the Tata binding protein, which has activated RNA polymerase, and it's going to start transcribing right here at the start site of transcription. Transcription factors can have their functions modulated by different mechanisms. One is binding of small effector molecules, such as a hormone. Hormones bind to receptors in the cytoplasm and bind to DNA responsive elements that turn on transcription. Transcription factors can also dimerize. They can form homodimers most often. Sometimes they can form heterodimers with 2 different polypeptides. And they need to form these heterodimers before they can bind to their elements on the DNA. Sometimes these transcription factors can be covalently modified by the addition of phosphate groups or other things. That process can either activate the transcription factors and activate gene expression, or sometimes you can inactivate transcription factors by that mechanism. What about steroid hormones? There's a large variety of steroid hormones in most mammalian systems and in other systems as well. There are estrogens, there are androgens, here we have progesterone, and a whole host of other hormones, cortisol. All of these hormones can change gene expression. Steroid hormones are produced by the endocrine glands and travel in the bloodstream and to their target cells and pass through the cell plasma membrane. They only stay in the cell, however, if they find receptors to which they can bind. And then they affect gene expression. So here's a cell, okay, here's a glucocorticoid. You're going to start right here with this glucocorticoid being produced by an endocrine gland, traveling through the bloodstream and zipping in and out of cells. If the cell has a glucocorticoid receptor, the glucocorticoid will bind. When the glucocorticoid binds, these receptors are modified, heat shock proteins are bumped off, and the glucocorticoid binds instead. That often reveals nuclear localization signals. These receptor proteins with the glucocorticoid bound to it dimerize, and they travel inside the nucleus and bind to DNA elements called glucocorticoid responsive elements, or GREs. Once they bind, they can turn genes off or they can turn genes on, either activate or repress gene transcription. So, in summary, general or basal transcription factors bind at the promoter and facilitate RNA polymerase binding. Some examples are the TATA binding protein and TF2D. In eukaryotes, regulatory transcription factors bind to DNA elements called silencers and enhancers. They are often at great distances from the promoters that they influence. Transcription factors have three types of domains: DNA binding, dimerization, and small effector molecule binding. DNA binding motifs include zinc fingers, leucine zippers, helix-turn-helix motifs. These domains can also sometimes function in dimerization. The mediator complex acts as a bridge between the activators and repressors bound at silencers and enhancers, and the general transcription factors such as TF2D and TATA binding protein that are bound at the promoter and to RNA polymerase itself. Transcription factors often form dimers, either homodimers or heterodimers, that increase their affinity for particular DNA binding sites. Transcription factors can bind small effector molecules that change their shape and consequently their affinity for DNA binding sites. Protein transcription factors can also be modified by adding phosphate, methyl, or sugar groups that alter their functional state. Steroid hormones pass from the bloodstream through the plasma membrane of the cell and bind to receptors in the cytoplasm. After some modification to expose nuclear localization signals, the receptor hormone complexes dimerize. They go into the nucleus, bind to GREs, and either induce or block transcription.